Before the pandemic, the lab of Stanford University biochemist Peter S. Kim focused on developing vaccines for HIV, Ebola and pandemic influenza. But, within days of closing their campus lab space as part of COVID-19 precautions, they turned their attention to a vaccine for SARS. Although the coronavirus was outside the lab's specific area of expertise, they and their collaborators have managed to construct and test a promising vaccine candidate. Our goal is to make a single-shot vaccine that does not require a cold chain for storage or transport. If we're successful at doing it well, it should be cheap too, said Kim, who is the Virginia and D.K. Ludwig Professor of Biochemistry. The target population for our vaccine is low- and middle-income countries. Their vaccine, detailed in a paper published January 5 in Axe Central Science. In addition to being the reason why these are called coronaviruses, corona is Latin for crown, these spikes facilitate infection by fusing to a host cell and creating a passageway for the viral genome to enter and hijack the cell's machinery to produce more viruses. The spikes can also be used as antigens, which means their presence in the body is what can trigger an immune response. Nanoparticle vaccines balance the effectiveness of viral-based vaccines with the safety and ease of production of subunit vaccines. Vaccines that use viruses to deliver the antigen are often more effective than vaccines that contain only isolated parts of a virus. However, they can take longer to produce, need to be refrigerated and are more likely to cause side effects. Nucleic acid vaccines, like the Pfizer and Moderna mRNA vaccines that have recently been authorized for emergency use by the FDA, are even faster to produce than nanoparticle vaccines. Initial tests in mice suggest that the Stanford nanoparticle vaccine could produce COVID-19 immunity after just one dose. The researchers are also hopeful that it could be stored at room temperature and are investigating whether it could be shipped and stored in a freeze-dried powder. By comparison, the vaccines that are farthest along in development in the United States all need to be stored at cold temperatures, ranging from approximately 8 to -70 degrees Celsius 46. This is really early stage and there is still lots of work to be done, said Abigail Powell, a former postdoctoral scholar in the Kim Lab and lead author of the paper. But we think it is a solid starting point for what could be a single-dose vaccine regimen that doesn't rely on using a virus to generate protective antibodies following vaccination. The researchers are continuing to improve and fine-tune their vaccine candidate, with the intention of moving it cl. Spikes and nanoparticles. The spike protein from SARS-CoV-2 is quite large, so scientists often formulate abridged versions that are simpler to make and easier to use. After closely examining the spike, Kim and his team chose to remove a section near the bottom. To complete their vaccine, they combined this shortened spike with nanoparticles of ferritin, an iron-containing protein, which has been previously tested in humans. Before the pandemic, Powell had been working with these nanoparticles to develop an Ebola vaccine. Together with scientists at the SLAC National Accelerator Laboratory, the researchers used cryo-electron microscopy to get a 3D image of the spike ferritin nanoparticles in order to confirm that they had the proper structure. For the mouse tests, the researchers compared their shortened spike nanoparticles to 4 other potentially useful variations, nanoparticles with full spikes or partial Testing the effectiveness of these vaccines against actual SARS-CoV-2 virus would have required the work to be done in a biosafety level 3 lab, so the researchers instead used a safer pseudo-coronavirus. The researchers determined the potential effectiveness of each vaccine by monitoring levels of neutralizing antibodies. Antibodies are blood proteins produced in response to antigens. Neutralizing antibodies are the specific subset of antibodies that actually act to prevent the virus from invading a host cell. After a single dose, the 2 nanoparticle vaccine candidates both resulted in neutralizing antibody levels at least twice as high as those seen in people. After a second dose, mice that had received the shortened spike nanoparticle vaccine had the highest levels of neutralizing antibodies. Looking back at this project, Powell, everybody had a lot of time and energy to devote to the same scientific problem, she said. It is a very unique scenario. I don't really expect I'll ever encounter that in my career again. What's happened in the past year is really fantastic, in terms of science coming to the fore and being able to produce multiple It normally takes a decade to make a vaccine, if you're even successful. In that case, the researchers are prepared to pivot again and pursue a more universal coronavirus vaccine to immunize against SARS-CoV-1, MERS, SARS-CoV-2 and future coronaviruses that are not yet known. Vaccines are one of the most profound achievements of biomedical research. They are an incredibly cost-effective way to protect people against disease and save lives, said Kim. This coronavirus vaccine is part of work we're already doing, developing vaccines that are historically difficult or impossible to develop, like an HIV vaccine, and I am glad that we're in a situation where we could potentially bring something to. A researcher from Chan Zuckerberg Biohub is also a co-author. Kim is a member of Stanford BioX, the Maternal Child Health Research Institute (MCHRI), and the Wu Tsai Neuroscience. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.